Good afternoon, everyone. So I'm really happy to introduce you Lauren Clance. Lauren is from Greensboro, North Carolina. She earned a bachelor's degree in biology with a marine science minor at University of North Carolina, Chapel Hill. Since June 2019, she's doing a master's degree at the Carmichael Lab, and she's now going to present us her research project on dolphins entitled Characterizing Site and Group Fidelity of Bottlenose Dolphins in Mobile Bay, Alabama, and adjacent Gulf of Mexico waters using photo identification and elemental analysis. Lauren, we are all ears.
Hello, everyone. Thank you to Debbrota for that introduction, and thank you all for coming to my presentation titled, as Debbrota said, Characterizing Site and Group Fidelity of Bottlenose Dolphins in Mobile Bay, Alabama, and adjacent Gulf of Mexico waters using photo identification and elemental analyses. My first chapter focuses on photo identification, and my second chapter focuses on elemental analyses, which will be broken down into stable isotope analysis and trace element analysis. Bottlenose dolphins are a globally distributed marine mammal species, so our research findings will apply far beyond coastal Alabama. They are known to inhabit diverse bodies of water, both coastal and offshore, including bays, estuaries, oceans, and rivers, many of which are highly impacted by human activities. Dolphins can also act as environmental sentinels, where their health reflects the health of the ecosystem they inhabit because they are long-lived and eat at high trophic levels. Currently, there are gaps in existing data for the northern Gulf of Mexico, including Alabama. Before the start of this project, little live animal work had been conducted in this region, and no photo identification studies had been conducted in Mobile Bay. This gap needs to be filled to establish baseline data for populations for future environmental perturbations, such as oil spills, harmful algal blooms, disease outbreak, or unusually high periods of freshwater discharge. Dolphins can be difficult to study because they are elusive, making it especially hard to research them at the population level. Photo identification, or photo ID, is an effective long-term monitoring technique to study them at larger scales. It is a mark recapture method where the mark is photographing individuals' dorsal fins, and the recapture is when they are resighted and photographed again. Dorsal fins are photographed because many dolphins have notches and markings that are unique to each individual, making fin patterns analogous to human fingerprints. Dolphins acquire these features throughout their lifetime, allowing scientists to identify them whenever they are sighted. By doing so, researchers can track movement patterns and define parameters such as site fidelity and group fidelity. Although movement patterns vary with geographic region and are thought to be driven by food availability, many studies have found that dolphins exhibit high site fidelity and in some cases may not leave areas with unfavorable conditions. This is the case, for example, with two studies conducted in Indian River Lagoon, Florida. Titcomb et al. 2015 found that dolphins in this area exhibit high site fidelity and group fidelity. Although there is some crossover between the communities, and each community here is outlined in a different color, they exhibited high site fidelity to their own respective segments. Mazzol et al. 2008 also found that dolphins in this region exhibit patterns of high site fidelity even when they're highly influenced by freshwater inputs and toxic metals. These graphics are from Cloyd et al. In Review and Cloyd et al. 2021, showing tag data for 2 of the 18 dolphins that were tagged off Dolphin Island during the 2018 health assessments. Each graphic is a different individual and shows movement patterns over a period of about 6 months. The animal on the left spent most of its time around Dolphin Island, while the animal on the right spent most of its time to the left of Dolphin Island. Both of these animals, including others in this study, exhibited high site fidelity to the region and had limited home ranges. My main objective for Chapter 1 is to determine if identified individuals are resighted in the same location, defined as site fidelity, with the same individuals, defined as group fidelity, in my study area, which is Mobile Bay, Alabama, and adjacent Gulf of Mexico waters. We conducted photo ID surveys in Mobile Bay in the winter of 2019 to 2020 and 2020 to 2021. We also conducted surveys in summer of 2020, and we will conduct surveys again in the summer of 2021. These surveys are boat-based and follow predetermined track lines pictured here, covering as much area as possible inside the bay and a contour line closer to shore to cover the perimeter. We follow these track lines at about 15 to 17 knots until dolphins are sighted, and we break off from the transect lines to collect data from this group. This data includes taking pictures of dorsal fins, noting behaviors such as feeding, socializing, traveling, and taking environmental data, which we collect with our flow-through system pictured here. Our data flow unit takes in water from the bay as we drive our transects and work dolphin groups. The water is brought in with the pump and transported to the case that contains probes for salinity, temperature, and dissolved oxygen, all of which are powered by the battery. After surveys are conducted, we sort photos by eye and add them to our catalog in FinBase. Photos are matched using a program called FinFinder, which utilizes machine learning to trace the trailing edge of dorsal fins, shown here in red, and it provides the top 50 matches from our catalog for that animal. From there, we determine if one of the given choices is a match or if the tracing is of a new fin. For my project, I'm only using photos from biopsied animals, which we'll discuss later. My hypotheses are that dolphins will exhibit group and site fidelity in the summer and winter. So we have our lovely blue, green, and gray dolphins here. And what this would look like is dolphins exhibiting high site fidelity to a portion of the bay, such as Bon Secours, and moving in these tight groups. Or dolphins could exhibit group fidelity, but not site fidelity regardless of season. So they would be moving freely throughout the bay, but still in the discrete social groups. Or dolphins will not exhibit group or site fidelity in any season and will move freely through our study area year round. Moving on to chapter 2, the first part covering stable isotopes. Stable isotopes is indicative of diet and shows trends with trophic levels. And they can be used as geographic tracers because they exhibit trends with salinity or source of productivity. So delta carbon 13 typically trends with salinity, and delta carbon 15 trends with trophic level. Looking at this graphic here, delta carbon 13 values typically become less depleted or less negative as you move from terrestrial to marine. And delta 15 nitrogen values typically become more enriched or more positive with increasing trophic level. So moving up along the y-axis here. So on the scale of the Gulf of Mexico, isotopic values in dolphin skin from live animals are distinct based on geographic location. We have Barataria Bay in purple, Mobile Bay in blue, and Sarasota Bay in yellow. Although there is some overlap between these two sites, all three areas still have distinct isotopic signatures. On a smaller scale, there are differences in abundance and diet off the coast of Alabama with shark populations, as found in Dryman et al. 2012. And even at this more confined scale of east and west, there were observable differences in isotopic signatures of animals that, like dolphins, are feeding at high trophic levels. Covering Alabama waters more widely, a similar trend holds here. So isotopic values in skin samples from stranded dolphins also showed geographic trends. And you can see the cluster of these values here. So Orange Beach is clustered here and is located at this location. You have Gulf Shores in gray, Dolphin Island in dark blue, and Mobile Bay in seafoam green, although it covers the entirety of Mobile Bay and not just this point. My first objective for chapter 2 is to determine if dolphins in our study area have distinct stable isotope ratios in skin that differ among spatially discrete groups. Because what dolphins eat should be reflected in their stable isotope signatures, we trawled for prey items that were available to dolphins at the time and locations that biopsy samples were taken. We trawled once each season at pre-selected sites and dissected muscle from prey items. So in the summertime, we have a single offshore site, a southern bay site, a mid-bay site, and a northern bay site. And in the winter, we had 2 offshore sites, a south bay site, a Bon Secours bay site, and a mid site. Dolphin skin samples were taken during remote biopsy surveys, where a modified crossbow is used to take a tissue plug from a 10 to 25 or a 10 by 25 or a 10 by 30 millimeter dart. This method allows researchers to take samples from dolphins without having to be hands-on. The picture here shows a dolphin that has been darted in the desired location on the body, outlined with this white circle, and you can see the sample in the dart here as it's rebounding. This picture shows what a typical biopsy sample looks like with a layer of skin and blubber. The skin and blubber are separated in the field and put in tubes inside a liquid nitrogen dewar. This map shows where biopsy samples have been taken, broken down by season. So we have summer in red and winter in green, as well as the trawl sites notified by the black dots here. We do have a good coverage of the bay with these samples. And additionally, we have 10 samples from training days in Mississippi Sound and around Dauphin Island that I will also be working up. Prey mussel and skin and dolphin skin both undergo the same process and are both being analyzed for carbon-13 and nitrogen-15. First, they are dried in an oven at 60 degrees for 48 hours. They're ground with a bead bug homogenizer, which is where you put your sample and beads in a tube, and they're shaken at 4,000 RPMs for 45 seconds, 5 times. The tubes are spun down to get the sample to the bottom, and the sample is packed in aluminum tins and sent to the UC Davis Stable Isotope Facility. If you're familiar with isotopes, dolphin skin samples would typically undergo a lipid extraction phase, but because my samples are too small for that, instead we will be applying a lipid correction factor as outlined in Cloyd et al. 2020. My hypotheses for the first part of chapter 2 are that dolphins in northern Mobile Bay will have more depleted delta carbon values than those in southern Mobile Bay. And this is because of that typical increase in salinity down bay. So we start off lower salinity in blue, and as we move to this bright yellow, salinity is increasing. And if you remember the figure from the introduction, those delta carbon values are increasing with the increasing salinity. Alternatively, if dolphins move freely throughout the bay, their isotopic signatures may not trend with salinity. Or if their prey items are highly mobile throughout the bay as well, this may be reflected in their signatures, even if the dolphins are not moving freely, and their delta carbon values may not trend with salinity either. Okay, so here's some preliminary data from the prey samples that show geographic trends with delta carbon values and trends with trophic level and delta nitrogen values. On the y-axis for both of these graphs is latitude, and on the x-axis here is delta carbon 13 values. Each of these clusters of points is associated with our site. So this group of points here comes from our first offshore site, this group of points with our second offshore site, this group of points with our southern site, this group of points with our Bon Secours Bay site, and this group of points with our mid site. And all of the colors are also representing prey items that are grouped by feeding strategy. So the black dots are anchovies, the purple dots are catfish, the orange dots are cephalopods, which is composed of squid. The gray dots are clubiforms, which includes fish such as shads, sardines, and menhaden. The decapod group in yellow is composed of shrimp and crabs. And the perciform group in blue contains fish such as drum, spot, and croaker. And what we're seeing is that carbon gets more positive or less depleted as you move north to south. So the carbon values are increasing along that increasing salinity gradient. To the graph on the right, the y-axis is still latitude, and the x-axis is those delta nitrogen values, where we would see that trophic level and nitrogen are increasing together. So here we have your decapod category that is typically a lower trophic level. Those have lower or less enriched nitrogen values, as opposed to groups such as the perciforms, which are higher trophic levels and have higher nitrogen values. We have the same setup here for the summertime. Each site is associated with a cluster of points. So here we have our offshore site, our southern site, our mid site, and our northern site. And we also have an additional prey group of flatfish, which contains bay whips and flounder. And the trends here are similar. Carbon is becoming less depleted as you move from north to south along that increasing salinity gradient. And we also see that lower trophic levels, such as decapods, have lower nitrogen values, and higher trophic level groups, such as perciforms, have higher nitrogen values. Moving on to trace elements for the second part of chapter 2. Trace elements can be natural or anthropogenic and have sources from places such as those in this diagram, from industry and agriculture, and also oil and the shipping industry, all associated with heavy metals. and freshwater discharge associated with elements such as strontium and barium. And all of these sources are prevalent in the Mobile Bay area or brought in from the Mobile Tensas River systems. Trace elements are necessary for biological processes, but can be toxic at high concentrations, and this is true for iron and copper. Some can also be both natural and anthropogenic, such as copper, which can be found of use in the shipping industry. Trace elements can also be used as geographic tracers like stable isotopes, because as you move away from a source, that elemental concentration is going to decrease. I'm going to be using trace elements in the context of geographic tracers, as well as indicators for contamination. Stavros et al. 2007 analyzed skin samples from live bottlenose dolphins in South Carolina and Florida. The trace elements associated with heavy metals and other kinds of anthropogenic contamination were found in dolphin skin, and these varied based on the two states. And it demonstrates that trace elements can be indicators of anthropogenic influences and used as geographic tracers. The case study of a stranded dolphin in Alabama and Deming et al. 2020 found that barium concentrations in the dolphin's teeth peaked around the same time as high inputs of freshwater into the area it was occupying, showing further evidence for trends of increasing barium concentrations with decreasing salinity. So the barium to calcium ratios here in black, they peaked at times of very low salinity here. My objective for chapter 2 is to determine if dolphins in our study area have distinct trace element concentrations in skin that differ among spatially discrete groups. To accomplish this, I conducted laser ablation inductively coupled plasma mass spectrometry, or LA-ICP-MS, and this method was chosen because biopsy samples mentioned previously are so small, and laser ablation is non-destructive. So by using this method, I had enough sample to conduct stable isotope analysis as well, which consumes sample. And it should be noted that the samples were dried before this analysis was conducted. Samples were analyzed for arsenic, barium, cadmium, cobalt, chromium, copper, iron, lead, and strontium. The laser portion of this instrument ablates or burns a small layer off the top of the sample, and the particles from this ablation are carried to the ICP-MS through these tubes with carrier gases. The plasma in the instrument ionizes the particles from the sample and strips them down to atoms so that the mass spectrometer can quantify amounts for each element. And finally, these measurements from the mass spec are sent to a computer file that I will be analyzing in the coming weeks using a software called IOLITE. So again, in general, the closer you are to the source, the higher your concentrations of that trace element or contamination will be. And Mobile Bay has several areas of input. We have agricultural runoff and freshwater that are brought down with riverine discharge, and the downtown area also has industrial and shipping activity. The west side of the bay has industrial centers as well, primarily near the Theodore Industrial Complex, and the eastern side of the bay has agricultural runoff near Fairhope. So I'm hypothesizing that dolphins in northern portions of Mobile Bay will have higher concentrations of trace elements associated with freshwater input and from these sources here, due to proximity to the riverine influence and presence of industrial centers on the mainland. I'm hypothesizing that dolphins on the western side of Mobile Bay will have higher concentrations of the trace elements associated with industrial centers as well, and that's due to the presence of them on the mainland and the Mobile Bay ship channel, which is nearby. And finally, I'm hypothesizing that concentrations of trace elements associated with freshwater and anthropogenic contaminants will be higher in the winter due to the increased riverine discharge, causing lower salinities and bringing anthropogenic runoff from upstream. Alternatively, if dolphins move freely throughout the bay, their trace element signatures may not be distinct, or there might not be enough resolution to distinguish spatially discrete groups. Implications for my research are that they're going to contribute to baseline data in the northern Gulf of Mexico by adding photos to our photo ID catalog in a larger catalog called GOMDIS that houses fins from dolphins on the Gulf Coast. The research will provide ongoing information about our live dolphin populations and allow for exchange of important data with the local stranding network, ALMMSN. It will give us a greater understanding of movement patterns and habitat usage, and it will give us a greater understanding of dolphin diet and what contaminants these dolphins are potentially exposed to. Now I will take any questions from the audience, and while you're thinking of those, I would like to thank my committee, Doctors Carmichael, Bovero, and Baker, our NOAA collaborators who provide heavy field assistance and heavy photoanalysis assistance, Carrie Sinclair, Kevin Barry, and Keith Mullen. Pretty much every single member of the Carmichael lab that provided heavy assistance in the field and help with presentation practice and just support in general. My funding source, Alabama Department of Conservation and Natural Resources, and of course, family and friends for their constant encouragement throughout my degree so far. Thank you for listening. Does anyone have any questions? I believe you may be able to unmute yourself or to type any questions in the chat box.
Hey, Lauren, it was a great presentation. Excellent job. One of my questions has to do with the fact that, so as we know, Mobile Bay is a highly stratified estuary in most of the year, pretty much because of the freshwater influx that is put in. Do you expect there to be any effect of that stratification on your isotope values that you're seeing, particularly because of the salt wedge and like the freshwater lenses that we get over a large portion of the bay?
Yeah, that's a great question. So certainly it could affect the isotope values, especially in the wintertime, where we seem to observe more bottom feeding by the dolphin populations, especially up north. So if they're in that salt wedge and their prey items are in that salt wedge as well, that could potentially affect isotopic values. But in general, over the winter, the isotopic and contaminant signatures could also just be a total wash because of how diluted the bay was and how quickly fresh water was moving through. Does that answer your question kind of?
Yes. Good job.
Thanks.
Hey, Lauren, this is Steve. Thanks for sharing. I was wondering how you're differentiating between winter and summer seasons, partially because winter, or we talk about high river discharge in the winter, but sometimes that doesn't start until much later in the season. And so kind of how are you separating those timing out and the discharge?
Yeah, our winter seasons that we surveyed are from December to February, and our summer seasons typically are from about June through August.
You might want to check the discharge at those times, because that could be a potential challenge in analyzing the data there, particularly like December and June.
Yeah, that's a fantastic idea. It's definitely something to look into, especially with our first winter sampling season of 2019 to 2020. We saw pretty low salinity values throughout the majority of Mobile Bay.
Hi, Lauren. That was a really nice presentation. I was just curious, some of the first isotope data I've looked at for the bay there, it looks like the nitrogen values were really quite a lot higher than I'm used to seeing. Do you have any comment on that?
That is a fantastic question. I would say, well, honestly, I'm not entirely sure, which is okay. Definitely something to learn and something to look into. Yeah, I am not sure. If anything, I would expect maybe some of the values to be lower if we're going to start bringing in some of the outputs from wastewater treatment plants. I mean, that may not propagate up high enough, but for higher nitrogen values than expected, I'm really not sure. For some of the higher trophic levels, like the clupiforms and the perciform group, that wouldn't be unexpected. But if you're also thinking that the decapod group has higher nitrogen values, then that's really interesting. It could be that the source that they're getting, the source of the productivity is more marine than you're used to seeing maybe.
Yeah, I think something like that's probably happening, or maybe agricultural input. I'm not sure. Anyway, great presentation.
Thank you. Oh, interesting. Greg Graber says that salinity was 2.5 PSU at the surface and 32.5 at the bottom on Tuesday over at the hole behind Fort Morgan. Yeah, those are definitely very large differences between surface and bottom. And all of our salinity readings are coming from the surface and not from the bottom.
I think one good point you've got there is the preliminary data you showed shows a nice gradient in carbon values down the bay anyway. Yeah, that was an issue I'd raised with you, but that data suggests that despite some of the complexities of salt wedges, you're still getting that resolution to distinguish upper from lower bay. So I think that looks pretty good.
Yeah, and the fact that we saw that during the winter time when there wasn't much different in salinity, or at least I felt like there wasn't from winter of 2019 to 2020. The bay was pretty low in terms of salinity throughout its entirety. It's exciting to see that those trends are still holding.
Lauren, do you want to talk just a little bit about, we have a 10-year data set essentially sampling at those same locations, and how your data during your sampling years compares to what we've found doing this for actually 10 years, that's 13-year data set.
Yeah, so interestingly enough, the trends that I found in that preliminary prey data are very similar and align very well with the data that's been collected over the past decade or so.
Hi everyone, good noon and welcome to my presentation. I'm Debbrota Mallick and currently working as graduate research assistant in Daphne Island Sea Lab, Isotope Geochemistry Lab under supervision of Dr. Wang. Today I'm going to present my prospectus, and the title of my prospectus is Chromium Isotope Systematics in Estuary Environment. Why we will study chromium isotope? Because the chromium isotope is an emerging redox proxy, and it can give us the important information about the Earth's oxygenation history. And exploring the Earth oxygenation history is important to understand the complex life on the Earth. Here we can see the figure is showing Here we can see the figure is showing the del 53 CR value from carbonate, rock, iron-rich sediment, black shale, and paleosol from the Archean Eon to Phanerozoic Eon. And when the atmospheric oxygen is increased from Archean Eon, especially from the great oxidation event to the neo-proterozoic oxidation event, the chromium isotope fractionated more positively. So this signature we can use to understand the atmospheric oxygenation level of the atmosphere, but this is a qualitative assessment of chromium isotope to understand the atmospheric oxygen. For quantitative assessment, we need a lot more study to understand that what range of atmospheric oxygen is correlated with which range of fractionation of chromium isotope. Chromium isotope usually expressed as del notion, which is the relative deviation of samples 53 CR and 52 CR to its standard material NBS 979 standard. For quantitative assessment, the global chromium isotope mass balance is important because the global chromium isotope mass balance is still poorly understood. And here we can see this is the current estimation of global chromium isotope mass balance. Here's the input is considered just only the river input, and after then from the ocean, chromium can output in different environmental setup. It is oxic environmental setup, hypoxic environmental setup, and the anoxic environmental setup in ocean. However, in this mass balance study, in this mass balance study, the input is only considered based on the river runoff. But estuary can also play an important role in this input to the in this shear chromium input to the ocean. So why the estuary environment is important? Because the estuary lies between the river and ocean, and it can affect the input of chromium to the ocean. And only few studies has been conducted in estuarine environment. I'm just going to backslide for a while, because here the current mass balance is showing that the input is only based on river input. So the study of chromium isotope study in estuarine environment also important to understand how the estuary is affecting the input of chromium to the ocean. My next interest is that the do sediment record seawater isotope signature. Understanding the oxygen evolution in the ancient ocean relies on sediment archives. That means how sediments is receiving seawater chromium isotope value and how it is different from oxic environment to anoxic to hypoxic environment. We need to understand if the sediment can reliably record the seawater del 53 value from different environment. There's not much studies done in this context, especially in sediment water boundary, which is considered as a transitional zone and can be very important that the chromium isotope is faithfully recording from the seawater to the sediment. Before going to details of my chapter, I want to give a short description about the elemental chromium. Chromium is a redox sensitive metal, has 4 stable isotopes, and there's a 2 valence state, trivalent and hexavalent, and both valence state, both chromium 3 and chromium 6 can stay in seawater. But hexavalent chromium is highly soluble and highly mobile, highly toxic in a certain concentration, and highly reactive. Trivalent chromium have low solubility, low mobility, and low or there's no toxicity, and it is highly stable. In a certain EAs and PAs, chromium can stay in seawater at different oxyanion species. The modern chromium biogeochemistry is related to the continental weathering of continental crust, continental weathering of chromium in presence of manganese dioxide and oxygen. When after oxidation, chromium 6 finds its way to the river, and then via estuary, it finds its final way to the seawater. In seawater, chromium can reduce back to the chromium 3, and as the chromium 3 is particle reactive, it can find its way to the sediment. From sediment, chromium 3 can reoxidize again and back to the water column. This is the general view of chromium, modern chromium biogeochemical cycle. My prospectus has 3 chapters. For chapter 1, I'm going to study chromium isotope fractionation in an anoxic estuary. For chapter 2, I'm going to study redox specific chromium isotope fractionation in Mobile Bay. For chapter 3, I'm going to study chromium isotope fractionation across the sediment-water interface. The redox-specific chromium isotope that I will study hexavalent chromium isotope and the trivalent chromium isotope differently. Is the chromium lost or gained in estuarine environment? Different studies have different conclusion in this question. Like here, the Sun et al. 2019 studied the chromium isotope from Connecticut River estuary, and they found that the chromium behaved non-conservatively in this estuary. The conservative behavior of an element is like that it is changing linearly with the salinity change. Here we can see the chromium concentration from lower salinity to higher salinity is not changing or increasing with increasing linearly. Similarly, del 53C are also not in a linear relationship with salinity. That we can say that in estuarine environment, the chromium can act as a non-conservative element. In contrast to the other study, Goring-Harcourt 2020 studied the chromium isotope from Bielu River estuary, and they found a conservative behavior of chromium isotope in this estuary. We can see and here in the figure that chromium concentration is changing very linearly with the salinity change from 0 to 30 PSU. And the del 53CR value also increased very linearly with very linear relationship with salinity from 0 to 30 ppt. So this indicates that the chromium behavior in this estuary is conservative. Why need to study the chromium isotope in anoxic estuary? So the ocean oxygen minimum zones are expanding, and the estuarine's seasonal hypoxia events also increasing, and it can really affect the behavior of chromium isotope in an estuarine environment. And there's not mass studies available, or there's not many studies done in this context, like in a estuarine anoxic environment. So that's for my first chapter. My key research question is that how does the chromium concentration and isotope change in the redox stratified water column of an anoxic estuary? My null hypothesis for this chapter is that chromium concentration and del 53CR behave conservatively in a seasonally redox stratified estuary. We have selected Greater Chesapeake Bay for our study because anoxia occur very annually in deeper water of central Chesapeake Bay, and it extends from May to September. We can see here in the figure, the middle bay is really very oxic, very anoxic, especially very anoxic, especially the bottom water. As the depth range from 0 to 40 meter, and about the lower bottom water is nearly very anoxic. And the dissolved oxygen concentration can range from 0 to 10 milligram per liter. So we have collected two batches of water column profile from this location of mid estuary where the bottom anoxia is present. After collection, we have separated chromium from seawater following the magnesium co-precipitation method. And after the magnesium co-precipitation method, the chromium plates with magnesium is collected from here, and then iron co-precipitation method was done. And after separation of chromium from the seawater, collected water, I ran the ion exchange chromatography, anion exchange chromatography, and the cation exchange chromatography to verify my chromium sample. And later, we ran through the mass spectrometry, Neptune mass spectrometry meter housed in Dauphin Island Sea Lab. And we measure both chromium concentration and isotope. Here we can see that our two data, the salinity for both of our data collected from our study area, salinity is increasing from surface to bottom, geo sharply decreasing from surface to bottom, and after about the 12-meter geo remain nearly zero. And the chromium concentration decreased from surface to bottom. Del 53 CR also decreased to surface to bottom. But which is this change of del 53 CR is a little bit different from other study, and del 53 should increase as the del 53 CR should increase as the DO is decreasing and the chromium concentration is decreasing, because as the chromium concentration is decreasing, which means the reduction of chromium 6 to chromium 6, chromium 3, and the chromium 3 is removing from the solution. If the lighter chromium 3 removed from the solution, del 53 CR value should be increased from surface to bottom. But we got the negative relationship with this thought, and this is the products of our study. For next step, we are going to use a redox-specific model to explain our data and to explain these products, that why our data is really different than what it should be. To solve this problem, and we also need the redox-specific data for chromium from natural water. We also need to measure hexavalent chromium and trivalent chromium isotope from natural water, and that is the focus of my next chapter. For chapter 2, the redox-specific chromium isotope fractionation in Mobile Bay. Redox-specific chromium isotope fraction can provide more information about the chromium isotope behavior in an estuary, and also it can explain that why chromium isotope is behaving differently, like our first study. And it can also solve these products. The key research question for my study is that why it is different between, what's the difference between the del 53 CR3 and the del 53 CR6, and how does the difference change with the salinity? For null hypothesis, and this chapter is there is no difference between the del 53 CR3 and the del 53 CR6 in oxic estuaries across the salinity gradient. For my study, I have collected 16 samples from upstream Mobile Bay to downstream of the bay with a salinity gradient from 0 ppt to 30 psu. All samples were collected from the surface within 1 meter of, less than 1 meter of depth. We found that the ratio of CR3 and CR6 is decreasing from 0 ppt salinity to 35 ppt salinity, and indicating that if we go from the upstream Mobile Bay to downstream Mobile Bay, the chromium 3 somehow will be lost in the estuary. That is also indicating that if we go from estuary to the broader ocean, CR3 will be lost. For chapter 3, I'm going to study the chromium isotope fractionation across the sediment-water interface, because the sediment-water interface plays a very important role for sediment biogeochemistry, and a little research has been conducted to investigate that how chromium concentration and chromium isotope composition is changing from seawater to sediment-water boundary, and how the del 53 CR value from seawater is recording in the sediment that we usually use to reconstruct the oxygenation history. So for this study, my key research question is that do sediment record seawater Delphi P3CR? And my null hypothesis is there is no fractionation of Delphi P3CR when chromium is removed from the seawater to sediment. For this chapter, I have not collected data, or I'm just now in the process of experimental setup and the field experimental setup. So we will follow the two research approach. The first is we need to conduct a lab incubation experiment, and later we will conduct a field experiment to test our laboratory experiments. It's a kind of validation study. So for our lab experiment, we will collect the sediment and will incubate it under different DO condition, and we will use artificial seed water for this study. And for this incubation experiment with different DO, we will measure LPP3CR from the water column and also from the sediment. As I already discussed that how we will, as I already discussed that how I did separate the chromium isotope from the water sample, I will follow the same procedure here to measure chromium isotope or separate chromium isotope from the water column. For sediment, we will follow the 3 steps to separate the chromium from the sediment. The first step is digestion of sediment sample using ASAP and nitric acid. And second step is about the ion exchange chromatography, very similar to my first study. And the third step is that analysis isotope using MC-ICP-MS, a mass spectrometry method. So here is a typical incubation chamber that I have collected from a very renowned study from Mobile Bay Kwon et al. 1996. And I'm going to follow this incubation technique in my study for lab experiment. And the expected result from the incubation experiment, I'm expecting from oxic water, the Delphi P3CR value will decrease from surface to bottom. And as it is the oxic water, oxygen will penetrate to the sediment within a certain depth, and within this oxic sediment area, Delphi P3CR value will be positive or will be fractionated positively. And from after the oxic sediment area, the Delphi P3CR value will be fractionated more negatively after the anoxic sediment boundary. For hypoxic water, I'm expecting the Delphi P3CR value will increase, and the sediment, a small layer of sediment will carry a little amount of dissolved oxygen as it is the hypoxic water, and the Delphi P3CR value will be stable in that area in sediment, and later it will reduce, or Delphi P3CR value will be more negative after the anoxic boundary. In anoxic water, I'm expecting Delphi P3CR value will increase from surface to bottom, and in sediment, Delphi P3CR value will be more negative than the hypoxic and oxic water. For field experiment, we will collect the sample, sediment sample, and the water sample from 3 different DO concentration. Like we can see here in the Mobile Bay, there's the extreme hypoxia in the upstream area, and the moderate hypoxia in the middle estuary area, and there is no hypoxia in the mouth of the estuary. So we will select these 3 regions for our study for oxic, anoxic, and hypoxic, depending on the DO concentration in these 3 proposed study area. For field experiment, we will conduct 9 experiments, which is about 3 salinity gradient, 0 PSU, 20 PSU, and 30 PSU, and the 3 environmental setup: oxic environment, hypoxic environment, and anoxic environment. For every experiment, we'll collect the water column sample, sediment, and also the pore water. But also for pore water, we'll only measure the chromium concentration, and for sediment, we will measure chromium isotope, and for water column, water sample, we will measure both concentration and isotope. Thank you very much to my committee members, and also the Wang Lab and the DSL Tech Service and fellow DSL colleagues for encouragement and supports. Thank you all. I can take questions.
Thanks, Dev, for your presentation. For others, I should mention that purifying chromium from the sediments and complex seawater is fairly complex, and Dev managed to learn the skills within 2 years since he started in 2019, which is very good. And at the end of the talk, if anyone has any questions, please unmute yourself or type in the text box.
Yeah, Dev, this is Jeff Krause. I'm curious about your Chesapeake Bay. It looks like there's some pretty strong salinity stratification. So are you just sampling 2 layers of water that are not really, a continuum, but just 2 separate mixtures? And if that's the case, then is there really a paradox?
We did not sample like 2 layers. We just collect the vertical column sample from the surface to bottom to investigate that how like the anoxia was going on. Probably there should be some stratification in surface to bottom water, but we did not collect samples separately from like surface layer or bottom layer. We just collect sample as usual as a column sample collection procedure. Yeah.